Hello friends, welcome back to another video of Cintas Medical World. This is me Cintas Sejo studying in University of Perpetual Health System Delta, Philippines. Today we are going to discuss about the basic unit of life, the cell. All the organisms are composed of cells. Cells are the functional and structural unit of each living being. The cells are too small that we cannot detect with our naked eyes, and it requires a microscope to see the cells. The study of cell is known as cell biology. We can mainly categorize the cells into two, the eukaryotic cell and the prokaryotic cell. Now we can see in detail about the eukaryotic and prokaryotic cell along with the cellular components or the cell organelles. There are two main divisions in the cell, that is eukaryotic cell and the prokaryotic cell. The eukaryotic cell is usually multicellular while prokaryotic cell is unicellular. The eukaryotic cell have a true nuclei, that means they are having a well-defined nucleus with a nuclear membrane. The prokaryotic cell does not contain a nucleus. In the eukaryotic cell, the genetic material is contained within this true nuclei, while in prokaryotic cell, the genetic material is not membrane bound. It is stored inside a region called the nucleoid. The eukaryotic cell contains organelles that is bound by membrane, while prokaryotic cell does not possess membrane bound organelles. Plant cell and animal cell is an example of eukaryotic cell, while the bacterial cell is an example of prokaryotic cell. Now we can discuss in detail about the cell organelles or the cellular components. The cell organelles are specialized structures that performs various jobs inside the cell. It is also known as little organs. The cell organelles are divided into membrane-bound organelles and non-membrane-bound organelles. The membrane-bound organelles is the organelles that is surrounded by a plasma membrane, while the non-membrane-bound organelles is the one that does not contain a plasma membrane. Examples of membrane-bound organelles includes lysosome, Golgi apparatus, endoplasmic reticulum, nucleus, mitochondria, chloroplast, vacuoles, etc. These organelles are embedded inside the cytoplasm. Cytoplasm is the jelly-like substance that is found in between the cell membrane and the nucleus. The examples for non-membrane bound organelles includes ribosomes, cytoskeleton, cell wall, etc. These are present both in prokaryotic as well as eukaryotic cells. Now we can see each of these organelles in detail. The first one is plasma membrane. It is also known as cell membrane or cytoplasmic membrane. The cell membrane is composed of lipid bilayer and proteins. One of the important functions of plasma membrane is to transport the molecules across it. Also, the plasma membrane is selectively permeable. That means it permits only the entry of selective materials in and out of the cell. In animal cells, the cell membrane provides shape and protects the inner contents of the cell. Then we have the nucleus. The nucleus is a double-membraned organelle that is found inside the eukaryotic cell. It is the control center of cellular activities and it is located inside the middle of the cell. It is the storehouse of DNA, the genetic material. There are pores in the nuclear membrane that allow the molecules to go in and out of the nucleus. Just like the cytoplasm, nucleus also contains nucleoplasm or the nuclear matrix. Inside the nucleoplasm, there is nucleolus and chromatin. There is also a spherical structure present inside the nucleoplasm known as nucleoli. It is responsible for the synthesis of protein and RNA. The endoplasmic reticulum is an organelle that contains network of tubules and flattened sacs. It helps in transporting materials throughout the cell. There are two different types of endoplasmic reticulum, rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes on their surface and it is involved in protein synthesis and secretion, while in smooth endoplasmic reticulum, there is no ribosomes present on their surfaces and it is the major site for synthesis of lipid. The Golgi apparatus is also known as Golgi complex. It is composed of flattened, stacked pouches called cisternae. It is responsible for transporting, modifying, packaging the proteins and lipids to targeted destinations. The Golgi apparatus or the Golgi complex is present in both animal and plant cell. The materials to be packaged is coming from the endoplasmic reticulum. Therefore, the Golgi apparatus remains in close association with the endoplasmic reticulum. The mitochondria is a double membrane bound sausage shaped organelle. It is the powerhouse of the cell. They produce energy in the form of ATP. It is also the site for aerobic respiration. The ribosomes are found in close association with the endoplasmic reticulum. 2/3 of ribosomes is composed of ribonucleic acid or the RNA and 1/3 is composed of proteins. It is not surrounded by any membrane, therefore ribosomes is present in both eukaryotic as well as prokaryotic cell. The ribosomes are named as 70S and 80S. The 70S is found in prokaryotes and the 80S is found in eukaryotes. The letter S refers to density and size. The S refers to the Swedberg's unit. 70S and 80S ribosomes are composed of two subunits. The subunits of 80S ribosomes are 60S and 40S. The subunits of 70S ribosomes are 50S and 30S. The primary function of the ribosomes is protein synthesis. The plastids are double membrane bound organelles. It is found in all the plant cells and euglenoids. Plastids contains pigments. Based on the type of pigments, the plastids are divided into three types: chloroplast, chromoplast and the leucoplast. Chloroplast contain chlorophyll and carotenoid pigments. These pigments help in trapping the light energy for photosynthesis. The chromoplast contains fat-soluble carotenoid pigments like carotene, xanthophylls, etc., which provides plants with their color like yellow, orange, red, etc. The leucoplast, they are colorless plastids which stores nutrients. Amyloplast stores carbohydrates. Elaioplast stores oil and fats. Aleuroplast stores proteins. The cilia and flagella. The cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures that cause movement of either the cell or the surrounding fluid, while the flagella is comparatively longer and is responsible for cell movement. Cilia and flagellum is covered by a plasma membrane. Their core or the inside part of it is called as axoneme. It contains 9 pairs of doublets of radially arranged peripheral microtubules. That means that there is 9 pairs of doublets of microtubules that is situated at a same distance from the center. And also there is a centrally located microtubules. So there is totally 9 pairs of doublets that is at the same distance from the center, and also a pair of microtubules that is located at the center. That makes the arrangement of the microtubules to be referred to as 9+2 array. The central tubules are connected by a bridge and they are embedded inside a part called as central sheath. Among the microtubular pair situated on the peripheral part. One of the microtubule is connected to the central sheath by a radial spoke. The peripheral doublets are also interconnected to each other by linkers. The cilia and flagella emerge from a centriole-like structure called basal bodies. The centrosome and the centrioles. Centrosome is an organelle that contains two mutually perpendicular structures known as centrioles. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are interconnected. Central part of the centriole is known as hub and it is proteinaceous. The hub connects the peripheral fibrils through radial spokes that is also made of proteins. During the cell division in animal cell, centrioles from the basal body of cilia or flagella give rise to spindle fibers. So we have found that the cilia and flagella is having 9 pairs of doublets of microtubules, while the centrosome and centrioles are having 9 evenly spaced fibrils of tubulin protein, and it is also triplet. The lysosomes. It is a membrane-bound sac-like structure. It is filled with hydrolytic enzymes. They have the capability to break down many types of biomolecules. Due to their function, they are known as suicide bags of the cell. They break down the cellular waste and debris by engulfing it with hydrolytic enzymes. The cytoskeleton is a network of filamentous proteinaceous structure that is present inside the cytoplasm. It is found in all the living cells, mainly eukaryotic cells. They provide a definite shape to the plant cell. The major functions of cytoskeleton involves mechanical support, motility and maintenance of the shape of the cell. The microbodies. These are membrane-bound, minute vesicular organelles found in both plant and animal cell. They contain enzymes and proteins. The vacuoles. The vacuole is a membrane-bound space that is found inside the cytoplasm. That space is filled with a fluid. They are bound by a single membrane called tonoplast. They help in the storage and disposal of various substances. They store the food or other nutrients required by cell to survive and also store the waste products. The vacuoles in the plant cells are larger than those in the animal cells. In the amoeba, contractile vacuole is important for the excretion. In protist, the food vacuoles are formed by engulfing the food particles. That is the end of today's video. I hope you guys understood the concept of the basic unit of life, the cell. Subscribe the channel for more videos. Like the video, share it to your friends, mention your valuable comments in the comment section. See you in the next video. Till then, it's a bye from Syntax Medical World.